Welcome! In this lesson, we'll build intuition for key gene regulation terms, letters A through T, and we will walk through two concrete matches with clear, visual models, allosteric enzyme and constitutive gene. Here is our A through T term bank. We'll refer back to it as we match. Match 1. The definition says, quote, binding at a regulatory site affects a catalytic site. That is the textbook behavior of an allosteric enzyme. Letter I. Let's visualize why. First, here is an enzyme with an active site where the substrate binds, and a regulatory site. When a regulator binds the regulatory site, the enzyme changes shape, which changes how the active site works. As a result, the substrate may bind better or worse. Match 2. The phrase says, quote, a gene that is expressed continuously with little or no regulation. That is a constitutive gene, letter A, always on. Here is a promoter with RNA polymerase sitting and transcribing over and over. Notice the repeat symbol. It keeps going without needing an activator or losing activity to a repressor. Now, a lightning glossary for the rest, each with a crisp picture. B. Enhancer, a DNA element that boosts transcription from far away. C. CpG island. C. Phosphate G rich region, often near promoters, frequently methylated or not in regulation. D. DNA methyltransferase, an enzyme that adds methyl groups to cytosine. E. Histone variant, a replacement histone that changes nucleosome behavior. F. Chromatin remodeling, nucleosomes slide or are repositioned to open or close DNA access. G. siRNA, small interfering RNA that guides cleavage of matching mRNA. H. Constitutive exon, an exon always kept during splicing. J. Polycistronic mRNA, one RNA with multiple protein coding regions, as in bacterial operons. K. Alternative splicing, different exon choices make different mRNAs. L. CAMP, a second messenger molecule. M. Riboswitch, an RNA that binds a small molecule and changes its own structure to regulate. N. Negative control, a repressor turns transcription down. O. Temperate phage, a virus that can be lysogenic, integrating into the host. P. Attenuation, translation of a leader influences transcription termination, like in tryptophan operon. Q. TFIID, general transcription factor complex, that recognizes the TATA box. R. Promoter bashing, mutating promoter segments to map functional parts. S. Operon, multiple genes under a single promoter in bacteria. T. Promoter, the DNA sequence where transcription begins. To summarize our two matches, 1. Binding at a regulatory site affects a catalytic site. That is an allosteric enzyme. Letter I. 2. A gene that is expressed continuously. That is a constitutive gene. Letter A. Great work!